In the name of Allah, the most beneficent, the most merciful. Worthy Vice Chancellor, University of Education, Professor Dr. Talat Nasir Pasha, respected Director, Division of Science and Technology, Professor Dr. Muhammad Alam Saeed, Chairperson, Department of Chemistry, University of Education, Professor Dr. Mia Habibur Rahman Mahmood, respected Speaker, Dr. Ishtiaq Ahmed Khan, dear faculty members and students, Assalamu alaikum. I am Dr. Muhammad Aslam, Associate Professor, Department of Chemistry, your host of the event. I welcome you all in the chemistry webinar series. Our today's topic is genomic surveillance of SARS-CoV-2 in Pakistan. I request you all to listen the talk carefully and ask questions if any comes to your mind. I welcome all of you again. To formally start the event, I would like to call Mr. Hafiz Usman Ahmed for the recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. هو الذي أنزل من السماء ماء لكم منه شراب من السماء ماء لكم منه شراب ومنه شجر فيه تسيمون ينبت لكم به الزرع والزيتون والنخيل والأعناب ومن كل الثمرات إن في ذلك لآية لقوم يتفكرون وسخر لكم الليل والنهار والشمس والقمر والنجوم مسخرات بأمره إن في ذلك لآيات لقوم يعقلون إن في ذلك لآيات لقوم يعقلون. अब इन्हीं आयात का तर्जुमा समाअत फरमाइए। पनाह मांगता हूँ अल्लाह की शैतान मरदूद के शर से। शुरू करता हूँ अल्लाह के नाम से जो बड़े मेहरबान निहायत रहम वाले हैं। वही तुम्हारे फ़ायदे के लिए आसमां से पानी बरसाता है जिसे तुम पीते भी हो और उसी से उगे हुए दरख्तों को तुम अपने जानवरों को चराते हो। उसी से वो तुम्हारे लिए खेती और जैतून और खजूर और अंगूर और हर किस्म के फल उगाता है। बेशक इन लोगों के लिए तो इसमें बड़ी निशानी है जो गौर व फिक्र करते हैं। उसी ने रात दिन और सूरज चांद को तुम्हारे लिए ताबे कर दिया है और सितारे भी उसी के हुक्म के मातहत हैं। यकीनन इसमें अक्लमंद लोगों के लिए कई एक निशानियां मौजूद हैं। सदक़ल्लाहुल अज़ीम।
Jazakallah. Now it's time to invite our speaker, Dr. Ishtiaq Ahmed Khan. Dr. Ishtiaq Ahmed Khan earned his PhD in the field of biochemistry in 2010 from International Center for Chemical and Biological Sciences, University of Karachi. He has completed his 2 postdoc training, one from Beijing Genomic Institute, China in 2012, and other from Wellcome Trust Sanger Institute, Cambridge, UK in 2015. He started his career as assistant professor at the Dr. Panjwani Center for Molecular Medicine and Drug Research, University of Karachi in 2012. Currently, he is working as associate professor and project director and in charge of Syn Forensic DNA and Serology Laboratory. He has published several papers in international, national reputed journals. He supervised establishment of the Halal Testing and Research Laboratory at the ICCBS and at the Kuwait Institute of Scientific Research. His group developed a new sensitive NGS-based method for detection of pork. His group is working on diseases genomic in Pakistani population during pandemic. His lab sequenced and reported around 200 whole genome from Pakistan. Now I would like to request Dr. Ishtiaq Ahmed Khan to deliver his talk on genomic surveillance of SARS-CoV-2 in Pakistan to the audience.
Bismillahirrahmanirrahim. Assalamu alaikum.
Walaikum Assalam, Dr. Will.
I am very thankful to Dr. Aslam, a very good friend, for extending invitation to present this very important talk. And I am thankful to Honorable Vice Chancellor, directors, students, and faculty members for joining us. As we know that SARS-CoV-2 is a pandemic of its kind. it is extensively said that humanity has never experienced the scale and the effect and impact of any pandemic so quickly. स्लाइड मूव नहीं कर रही आप let me check. आप लाइन पे रहेंगे हम बताइए So SARS-CoV-2, as I told, that it is a pandemic of its kind, and it spread very quickly, and it appears that SARS-CoV-2 caught the humanity unprepared. But this is not the case. Things were very much known, but due to certain reasons, world was not prepared for that. Coronaviruses are not unfamiliar with humans. We are very familiar with coronaviruses. There are around 200 known coronaviruses. Most of them are not associated with any disease and they are not pathogenic. So, coronaviruses were introduced to humans as a disease-causing agent first time globally in 2002 to 2004 when severe acute respiratory syndrome outbreak was experienced in China, and then Middle East respiratory syndrome. The first case was identified in June 2012 in Jeddah, Saudi Arabia, and most of the cases occurred in Arabian Peninsula. So the SARS-CoV-2, which we are going to discuss today, belongs to these two pathogenic coronaviruses like SARS and MERS. Before moving forward, I would like to draw your attention to this very important paper. There are lots of conspiracy theories about the origin of SARS-CoV-2. Some people say that it is prepared in laboratory and then purposely it is spread for some political reasons. Another point is that which conspiracy theorists, they usually present that SARS-CoV-2, it was prepared in Wuhan, China, Institute of Virology, and then it is distributed, spread to the other part of the world. But interestingly, a paper was published in November 2015, and if you can see the funding agency, National Institute of Health was the funding agency of this work, and key laboratory of special pathogens and biosafety, Wuhan Institute of Virology, Chinese Academy of Sciences, and Department of Epidemiology, University of North Carolina, USA, were the key partners. So this study is about SARS-like clusters which are circulating in the Chinese bird populations. So just see few punchline of these articles. It says we generated and characterized a chimeric virus expressing the spike of bat coronavirus SHCO14 in a mouse adopted SARS-CoV backbone, and the result indicate that it can infect the mice through SARS receptor human antigen converting enzyme 2. This virus, which was basically prepared in the laboratory by the two collaborating institutions, one from China and one from USA, it infected the human cells and the mice cells with almost same efficiency as SARS-CoV-2, and the same receptor was used, which is human antigen angiotensin converting enzyme 2, which is basically the receptor for the SARS-CoV-2. Then see the second line. Additionally, in vivo experiment demonstrate replications of the chimeric virus in mouse lung with notable pathogenesis. So this is being done in 2015. Then evaluation of available SARS-based immune therapeutic and other medications revealed poor efficacy. Both monoclonal antibodies and vaccine approaches failed to neutralize and protect from infection with COVs using the novel spike protein. So again, it was evaluated that this virus will be not managed with the available therapies and the SARS-based vaccines. And at the end, they suggest that a potential risk of SARS-CoV re-emergence from viruses currently circulating in bat population is there. So, in summary, I would say that in 2015, we were very much aware of the fact that a pandemic is very near, and the cause of this will be likely a SARS-based virus. So alarm bells were ringing. You can see another paper 2 years before the emergence of this pandemic in 2017, understanding bat SARS-like coronaviruses for the preparation of future coronavirus outbreaks. So, as I am telling that we were very much aware of the fact that SARS-CoV-2, the emergence of SARS-like virus is very likely, and it can get out of control. So, according to Johns Hopkins University Corona map, up to now there are 247 million cases and about 5 million deaths are confirmed worldwide. In Pakistan, there are 1.2 million confirmed cases and more than 28,000 deaths with 1.222 million recoveries. So, as per our understanding and the observation, the actual caseload in Pakistan is many-fold higher than the confirmed cases. So here you can see the map of confirmed cases in Pakistan. So there are around 23,000 confirmed cases in Balochistan, which are the least numbers. So SARS-CoV-2 genome is around 30,000 nucleotide long, and the Wuhan reference was 29,903, around 30,000. It is a positive sense single-strand RNA virus, and it replicates to synthesize its copies. SARS-CoV-2 attaches to the ACE2 receptor, which is present in type 2 alveolar cells, bronchial epithelial cells. They are present in tongue, buccal epithelia. They are present in myocardial cells, kidney proximal tubule cells, and so on. So SARS-CoV-2 binds to ACE receptor 10 to 20 times strongly than SARS-CoV-1. So SARS-CoV-1, which was basically causative agent of the SARS disease, SARS outbreak, SARS and MERS, these two diseases which were previously experienced were more, lethal than that of the SARS-CoV-2. But the point is that SARS-CoV-2 is highly infective, and that's why it causes that much damage. So the virus enters the cell through S protein, spike protein, I think one of the most famous proteins right now. S1 subunit attaches with ACE2 receptor for binding, and S protein is then cleaved by using cellular serine protease TMPRSS2, and this allows fusion of the viral and cellular membranes, and eventually the virus enters in the cell. As well as the pathogenesis of SARS-CoV-2 is concerned, there are two important points to be taken into consideration. Number one, we know that interferon-based system is one of the first defenses for the any viral infections. So SARS-CoV-2 basically undermines the interferon-based immune system, immune response, and it overexpresses different genes which eventually result in the cytokine storm, and then it results in the immune cell suppressions like T follicular cells and B cells, they are suppressed, and the antibody response is also suppressed. So on one hand, the immune response is muted, and on the other hand, cytokine storms damage different organs, and it can get out of control. So you can see here that in serious cases, SARS-CoV-2 can damage any of the tissues in the body. It can impact on brain, eyes, nose, lungs, heart, blood vessels, and livers. So patient usually experience different type of serious patients, especially they experience severe type of damages to different tissues. The severity of the disease also depends on the strength of the immune system and genetic of the host. So that's why its effect on infected individuals are very diverse. It ranges from asymptomatic to lethal. Most of the time, we think that SARS-CoV-2 is a disease which comes and goes, but this is not the case. NIH, National Institute of Health USA, has launched a new initiative to study the long COVID. So in many patients, it is said that around 25% patients which have severe symptoms after infection of COVID, they experience long-term effect of COVID from memory loss to repeated lung problems to kidney failure and so on. So here you can see this is a study published in Scientific Reports in August 2021, and you can see here that how many different types of impacts are being faced by the recovered patients, and these are the long-term effects. So 80% of the people which are recovered from the COVID after having symptomatic COVID, they experience at least one of the following symptoms. These symptoms can range from chills to pains to weight loss to headache, attention disorders, anxiety, depression, fever, and so on. So right now, the COVID as a pandemic is going down, but a big challenge persists, and that is the management of the long-term effects of the COVID-19 on the patients. vaccination is a safer way to build protection, and it is one of the most important way to get rid of this pandemic. There are, a range of vaccines available, and COVID vaccination will help to protect by building immunity without the risk of severe illness. Right now, more than 7 billion doses of vaccines have been administered worldwide. In Pakistan, 105 million doses have been administered, around 71 million doses as first dose, and there are 41 million people which have gotten both doses. So this is the share of people vaccinated against COVID-19 till October 24, 2021. So you can see that different countries have a very high proportion of the vaccinated people, like UAE, Portugal, Cuba, Chile, Spain, and so on. Pakistan has a long way to go still. Before moving forward, I would like to just briefly describe how the vaccines are developed. There are different vaccine platforms, and vaccines can be of inactivated viruses, live attenuated viruses, viral vector vaccines are there. There are some vaccines which are based on virus-like particles, then recombinant subunit-based viruses, vaccines, and most recently, first time mRNA-based vaccines have been introduced. Pfizer vaccine is mRNA-based vaccine. So here you can see different vaccine platforms like Moderna and Pfizer have developed the messenger RNA-based vaccine, which are highly efficient, like BNT162b2, which is developed by Pfizer, is 95% efficient. Moderna is 94.1% efficient. And then comes the non-replicating viral vectors. They have a range from 70 to 50% of efficiency. And then protein subunit-based vaccine, they are also quite effective. So a range of vaccines based on different methods and different technologies have been developed. Sputnik 5 was the first vaccine which was prepared in Russia, and it was first administered. Initially, there were much doubts on the effectiveness and safety of the Sputnik COVID vaccine, but a study published in Nature in July 2021 clearly mentions that there are mounting evidence which suggests that Sputnik COVID vaccine is safe and it is effective. Another very important point is the effectiveness of vaccines. There is a serious concern that because vaccines, most of the vaccines, or you can say almost all of the vaccines, they got emergency approval, and lots of data which is normally required for the approval from the regulators was missing when vaccines were approved. So, but most of the vaccines, they proved safe. But there is major concern that the immunity which is produced by these vaccines is waning. So lots of studies are going on, and this is the latest one which is published in this October, that some people, it is observed that they are having super immunity and they are immune against almost all of the variants, and they are not affected. So it is being investigated that what is behind that, that why some people are super immune and others are not that much. You have seen the people which are being infected by the SARS-CoV-2 twice or thrice, And there are cases of vaccine breakthrough that vaccinated people are also being infected. This super immunity, which protects against all the different variants, even more infectious variants, interestingly, it comes with the first natural infection. So, in the nutshell, it is deduced from this study that if a person is previously uh, infected with SARS-CoV-2 by natural infection, and then after recovery, it is the person is administered with both doses of vaccines, any of the vaccines. So his or her immunity will be a kind of super immunity. So it is also observed that in the people which have infection, normal nature infection, and then they are vaccinated, their antibodies are diversifying even after one complete year. But the antibodies which are made under the effect of vaccination, 2 doses of vaccination, they stop diversifying after 2-3 weeks. So it means that there will be a chance that the people who are infected, who are not infected and got vaccinated, they will be infected with future variants, and they will probably require booster doses. Viruses rapidly evolve during pandemic. You know that it's all about numbers. When viruses infect more and more people, like in case of SARS-CoV-2, hundreds of millions of people have been infected, and so the virus divide billions and billions and trillions of time. So in this case, they rapidly evolve. This contribute to the viral adaptation, vaccine escape, drug resistance, and higher transmissibility. There are many descendants of the original Wuhan strain, and it have been already evolved in many lineages. A massive genome sequencing drive is underway globally, and more than 1 million genomes have been sequenced and reported. Most of the genome sequencing efforts have been done in UK. UK is at the top of this effort, and then US and other countries. Developing countries, they have a little contribution in the genomics. Spike protein, as we all know that through spike protein, SARS-CoV-2 interacts with the cells and it eventually enters in the cell. So it is highly associated with the infectivity and antigenicity of the SARS-CoV-2. So most of the, you can say almost all of the variants, more infectious variants like alpha, beta, delta, gamma, epsilon variants, they have mutation in the SARS-CoV-2 spike protein gene, and especially in the receptor binding domain of the spike protein. So here you can see that this is the impact of SARS-CoV-2 variants on vaccine efficacy and effectiveness. So in summary, I would say that most of the variants, like especially the beta variant, which was emerged from the Brazil, from South Africa, it decreases the efficacy of the vaccines to a higher extent. Like in case of AstraZeneca, against Wuhan strain, the efficacy of AstraZeneca is from 55 to 81%, but against beta strain, which was emerged from South Africa and previously it was called as South African strain, it is just 10%. Genomic surveillance is a strategy for the detection and control of the more infectious variants for the prevention and disease control. And the finding of such studies support contact tracing, social distancing, and travel restrictions to contain the spread of SARS-CoV-2. But we have seen that different variants like Beta variant emerged from South Africa, and Delta variant from India, and UK variant from UK. So these variants emerged. Sometimes they were timely detected. Lots of travel restrictions, social distancing, and other measures were taken. But those measures, because the world is a global village, and those measures almost remain ineffective, and the world remain unable to counter the spread of like Delta variant, which was emerged from India and proved to be very devastating. So based on emerging and recurrent mutations, the viruses, there are two types of mutations. Virus keep on mutating, and sometimes they have some mutations which make the viruses unfit, and these mutations don't go down in further lineages. But there are several mutations which are either have no effect on the fitness of the virus, or they make virus fitter and more infectious. So such variations which make viruses more infectious, more proliferative and more fitter, they then go down in different lineages. So there are two main lineages, or you can say types of the SARS-CoV-2, which are A and B. There are other lineages, but A and B are most important. A is now largely suppressed, and it has roots from the Wuhan, China lineage. It is that basic lineage. And B lineage, base of this lineage lies in China with many global exports. And this B lineage is identified by two distinct single nucleotide polymorphisms, which is T8782C and C228144T. So here you can see the distribution of SARS-CoV-2 lineage. You can see that most of the time this is the B lineage which have spread around the globe and presently persisting in different parts of the world. As I discussed earlier, that in pandemic especially, the viruses, they keep on mutating, they diversify. The more infectious ones are positively selected, and then they keep on infecting till there is an effective immunization through vaccination, or there is a kind of herd immunity that most of the people are infected, and then they are naturally immunized. So here you can see this is an important slide which demonstrates that how different variants of concerns and variants of interests are distributed and how they are emerged. Like UK variant, a very famous variant, as I told you that most of these variants belong to B lineage, and almost exclusively all the variants of concerns and variants of interest, they belong to the B lineage. So you can see here on the y-axis, these are the different regions of the spike protein. This is N-terminal domain, receptor binding domain, and you can see that many variants of concerns and variant of interests lies in the receptor binding domain. L452R and Q, this is a very important mutation, which is also present in the Delta variant. So B lineage is distributed in Asia Pacific, and so on. There are concerns that viral variants, they can re-emerge into a very, very bad new wave. This concern is due to the fact that there are, some countries which are very resourceful and they have a very effective genomic surveillance-based system, and they can quickly identify new emerging and more infectious variants. Like a few days back, there was a news that the scientists in UK, they are continuously monitoring the emergence of new variations in Delta variant, and those variations are making Delta variant even more infectious. So the problem is that in developing countries, the vaccination is not that good, and there is almost, in many countries, there is no genomic surveillance-based system. So in those countries, the healthcare system and the governments are unable to identify and counter the new emerging variants. So it is a threat that probably there are some more infectious and dangerous coronavirus variants which are being emerged in those countries where vaccination is not that good, and where there is not a good and effective surveillance system. So in those countries and from those countries, new variants can emerge unnoticed, and they can spread in the world and overwhelm the world again. So the take home is that an effective and a global effort for monitoring of these variants through genomic surveillance is needed. We at International Center for Chemical and Biological Sciences started the genome sequencing efforts since the start of the pandemic. We were the first one who sequenced the complete SARS-CoV genome within Pakistan, and these are the first 3 strains which were sequenced from Pakistan. We sequenced in-house, and other were sent abroad for sequencing. And in these 3 strains, there was no mutation in the spike proteins. Mutations in spike proteins, they emerged later. So far we have sequenced more than 200 genomes. So far we have sequenced more than 200 genomes at the Jamilur Rahman Center for Genomic Research in a spatial-temporal fashion. We collected samples from different hotspots of Pakistan, and these areas include Karachi, Lahore, Quetta, Murdan, Peshawar, and Islamabad. I am skipping these slides. These are largely about methodologies. We use library construction workflows of Illumina and run the sequencing libraries on the Illumina-based MySeq system. Illumina MySeq system is basically sequencing by synthesis-based technique, and it is being used globally. During the first wave, we sequenced around 48 complete genomes, and we mapped those genomes with the reference genomes from Wuhan, and we identified 122 mutations, and there were 74 singletons and 48 recurrent mutations. Recurrent mutations are those which are emerged in more than 1 samples, and these are basically the mutations of interest, and these mutations are then monitored that either these are being spread in larger populations. During the first wave, we identified the mutations which were highly recurrent for D61G, and most of our variants were belonging to the B lineage. So this is the mutation allele frequency spectrum. On the x-axis, you can see allele counts, and on the y-axis, number of mutations. So you can see that larger number of mutations were singletons, but there were several recurrent mutations. Up to 33 samples, we found some mutations which were that much recurrent. So from May to August in 2020, we observed that the mutation rate per genome gradually increased, and genetically the genomes got more and more diversified. We also observed the decrease in singleton and increase in the recurrent mutations. Here you can see that mutation rates is mentioned in 4 genes during 4 months from May to August 2020, and you can see that mutations in spike proteins have been emerged. So this is the mutation landscape. Different colors, they show different types of mutations, nonsense mutations, frame shift mutations, and mutations present in the 5' and 3' UTR regions. So you can see that most of the mutations which were identified in the spike protein are missense mutations. One important point which I would like to mention is that these mutations, large number of mutations which were observed, only few have been experimentally characterized and their functional impact have been studied. So this B614G is one of those mutations on which functional studies have been carried out, and it is proved that this B614G mutation makes it more infectious, and that's why it spread globally. This image shows the migration routes, that how different strains are introduced in Pakistan. So you can see here different countries like Saudi Arabia, Iran, UK. These are the countries from where most of the mutations were introduced during the first wave. There are different Middle Eastern countries. You can see here France as well, Italy, and so on. So this is the phylogenetic analysis of the different strains which were identified during the first wave, and you can see that mostly we found B.1.36 and B.1.160 lineages. B.1.255 lineage was also identified in few cases. Mutations identified during second wave. So we observed P681 mutation, which was also identified in UK strain, and L18F, but we didn't identify any UK variant at that time. So these mutations, which are P681H and L18F, these mutations are also present in UK and South African strains, but these were emerged independently in our strains because when we assign a strain based on the sequencing, so it is based on the haplotype. There is a group of mutations which represent a specific strain like UK and South African variant. This is a comparison of mutations which were identified during first and second wave. And these are the mutations in 3 structural proteins like spike protein, membrane protein, and nucleocapsid protein. So you can see that more than 80 mutations were identified in the spike protein. So lineage of second wave samples, B136. This is very interesting. B136 was the strain or lineage, you can see, which was being observed in Pakistani population before the introduction of UK variant and Delta variant. So it clearly implies that probably in our own condition and in our own genetic landscape, B136 was being spread more visibly, but it was wiped out after the introduction of UK and Delta variant. During the third wave, we sequenced 26 whole genomes and we identified 247 mutations, and 59 mutations were shared with the first wave. So during the third wave, most of the variants which we identified were UK variants, and this was the genomics based and sequencing based surveillance was only the part of our efforts. When we were able to identify different strains based on the whole genome sequencing, we also used RT-PCR based assay extensively, and we identified thousands of variants based on the RT-PCR assays. But where it was necessary, we used the sequencing to confirm the different strains. During fourth wave, whole genome sequencing of 90 samples were carried out, and samples were collected from July to September 2021. 487 mutations, several of them were present in spike protein, were identified, and these mutations which were identified during the fourth wave include 338 singletons and 149 recurrent mutations. We are continuously monitoring these mutations and their impact on the overall spread of the virus. Among the highly recurrent mutations, L452R and 478K were detected, which were present in the receptor binding domain of the spike protein, and these two mutations are designated as the variants of concern according to the WHO classification. And these are the characteristic mutations of Delta variant. So most of the samples, around 80 samples out of 90 belong to Delta variant, but still there are a few variants belong to other strain like B136 lineage. They are still present. So interestingly, in the second wave, in the absence of UK and Delta variant, B136 lineage was persisting in our population, and even after the introduction of more infectious Delta variant, lineage B136 still persists. This is the allele count. This is the comparison of different mutations in spike protein in different during 4 waves, first, second, third, and fourth wave. And most of the mutations in spike proteins were identified during the fourth wave. So I am thankful to Professor Dr. Taur Rahman, Veteran and Chief of International Center for Chemical and Biological Sciences, because the center where we are currently working, Jamil Rahman Center for Genome Research, was initially established by the personal donation of Professor Taur Rahman. Our director, Professor Dr. Muhammad Iqbal Chaudhary, he is also Coordinator General of the CONSTEC. He is very supportive. My colleagues, Dr. Muhammad Shakil, Dr. Muhammad Irfan, they carried out most of the experimental and analytical work. Dr. Muhammad Rashid from the biology center, he was also involved in the study. Zaidun Nisa, my PhD student, is also involved. Our PhD project is on the genomic surveillance of SARS-CoV-2 in Pakistan. Dr. Simon Siddiqui from Institute of Biotechnology and Genetic Engineering, Islamabad, and Dr. Noorulain, and Dr. Shah Jahan from University of Health Sciences, Lahore, and Dr. Nematullah from Balochistan. They all were involved in discussion, planning of these experiments, and providing the samples. So thank you very much for your attention.
Thank you, Dr. Shah, for such an informative talk. The forum is now open for questions.
Why Pakistani people were not much affected by COVID-19?
Hello.
Ji, sir. Why Pakistani people were not much affected by COVID-19?
This is a very good question. Lots of people, they ask that why Pakistani population. I think our National Command and Control Center, NCOC, very actively took measures and very timely measures. And I think it was a team effort from our medical professionals, from our government institutions, and coordination from the NCOC. So I think smart lockdown policy, it proved to be effective. Our educational institutions were timely closed. Time to time, lockdown were also imposed. Uh, but above all, there are still things which are not much explainable. Because our genetics is like the genetics of Indian people. So we were much feared after the introduction of Delta variant that what kind of impact it will have on our population. But by the grace of Allah Almighty, it was nicely managed. But I would give the credit to the NCOC, the very effective coordinated measures. They make sure that our medical legal, our medical professionals, they get personal protective equipment, timely vaccination efforts were timely started, and the government threw much resources to counter this. So this is a teamwork. This is the result of collaboration and coordination between different institutions. So behind the scenes, there are lots of efforts which resulted in this result. And the response of Pakistan is one of the best responses against the COVID-19 in the world.
Okay, sir.
Assalamu alaikum, sir. First of all, thank you so much. It was very informative. And now my question is that, sir, is the new strain of COVID-19 virus more leading to complication and mortality as compared to wild type or not?
As well as beta is concerned, beta lineage is concerned, it was infecting children as well. We noticed many children in Karachi suffering from COVID-19, and they went to complications, and they even suffered from pneumonia. So the other variants, their pathogenicity is almost similar to that of the Wuhan. They are more infectious. So in pandemic, being more infectious means it will be more dangerous, because for instance, if the mortality rate is 2% for a strain which can infect 2,000 people in one day, and compared to a strain which can infect 1 million people in 2 days, for example. So even if the mortality rate is 2%, more infectious will appear as more lethal. So no doubt, more infectious strains will have worse overall impact on the infected population.
Okay, sir. Thank you so much. And sir, my second question is that, what kind of evolution can we expect from coronavirus in the recent future?
As I told you that there are mutations which are characterized, but the number of those mutations which are functionally characterized is not that much. We can estimate the positive selection of variants based on their recurrence and their prevalence. So if one mutation is appearing in more and more variant, in more and more people, so it clearly implies that it has some advantage for the virus. It may be helpful in infection or proliferation of the virus or overall fitness of the virus. But if a variant like L452R variation, for example, or D614G variation, so these variations are highly recurrent mutations. So when they are there and they are highly recurrent, it means that they have some advantage for the overall fitness of the virus. So, but very few mutations have been directly characterized.
Okay, sir. Thank you.
Assalamu alaikum warahmatullahi wabarakatuh. MashaAllah, Inspector sir, the presentation was too much impressive. It cleared much queries related to this pandemic disease. Inspector sir, I want to ask that what are the important targeted sites of coronaviruses, like related to age, region, or profession in the whole world?
Yeah, age and other diseases like hypertension, diabetes, hepatitis C, uh, aging, these are the risk factors. So, when vaccination was started, so preference was given to the aged people and people who are suffering from some other complications and diseases. So these are the risk factors. Yes.
One more question. What do you suggest in future for such type of pandemic diseases, or what control measures should be taken by the WHO?
This is a very good question. Basically, first thing is knowledge, as well as we learned a bigger lesson from SARS-CoV-2, and the lesson is that uh, the viruses which infect and which spread through the respiratory system, they need to be identified. So the point is that there must be more investment in virology research, and uh, coronaviruses and other related viruses, or any other virus which may have a potential to infect humans, they must be investigated. And if we know about the potential of a virus, so we can take measures to counter that. So the first point is that there must be more investment in research to identify new types of potential viruses which can potential candidates for the pandemics. And second lesson is that there must be coordination, larger coordination among different countries on international forums and inside the country from different departments. You know, uh, pandemics, world is a global village. So if there is an infectious disease which have a potential to spread around the globe, so the world is a global village. So there are lots of communication. People are traveling, goods are being transported. So there is no chance that one could build walls around its own country or around its own nation and could spare from such pandemics. So it's not possible. So overall coordination is required. And third, uh, the future pandemic may emerge because that the advanced countries, they are tightening their grip on research, and they are more vigilant. Their viral and pathogenic surveillance systems are getting more and more advanced. But probably the future pandemic may emerge from underdeveloped countries, poor nations, where things are evolving and going unnoticed. So, an international effort must be there to initiate uh, the uplifting of the healthcare system in the more poorer nations and developed countries, because if there will be something like pandemic originating from the poor countries, for example, it will not spare the advanced nations. So all the world will bear the brunt, like it has seen in the case of SARS-CoV-2. So these kind of measures, I think, can be taken, and WHO and other international organizations, they are seriously considering such measures.
Jazakallah, Inspector sir.
Audience, if there is no question from you, then I would like to request Dr. Misbah Shah, Associate Professor, Department of Chemistry, University of Education, for valuable comments on this webinar.
Thanks so much, Dr. Aslam, for nice introduction. Dr. Aslam, can you hear me?
Yes.
Okay. First, mic was not working. Thanks so much, Dr. Ishtiaq Ahmed, for such a valuable and informative talk about COVID-19 and gene sequencing. In fact, COVID-19 has posed a lot of challenges, keeping in view the social and economic values. It has emphasized the true value of identifying the business strength and weakness, as well as recognizing and mitigating the material risk as well. And after COVID-19, like in last 2 years, there are important outcomes about it. First one is the learning curve for the COVID-19. It's steep and it is relying on the data and the evidences which are based, policy based, and making is paramount to the saving lives. And the second outcome is the decision which must be based on an integrated framework of top to bottom and bottom to up policy making. And it's also pertained to the investment in the neglected public health sector through sustainable programs. And fourthly, public engagement through effective government messaging is instrumental. And lastly, it also enforced and it has provided us to overview the how to cope with such type of the disasters. And therefore, the humanity has learned how to adopt disaster preparedness and build resilience across the entire governance of the structure of the certain country. And therefore, I will conclude that yes, this COVID-19 has changed our life, frameworks of our life, our infrastructure, our way of learning, communication. It has changed in a huge background. And therefore, now it's need of the hour. Yes, we have to go with this disease. We have to save it, but still we have to do the research work in our labs, and we have to go in the stream along with the other country. We should be progressive as well. Therefore, I will thankful to organizing team, Dr. Muhammad Aslam, and our valuable speaker, Dr. Ishtiaq Ahmed, for presenting such an informative session, which has given us very information, not about the COVID, but overall about the whole world as well. The data and the statistics they are quite interesting and informative. Thanks so much. Thanks so much, Dr. Aslam.
Welcome, Dr. Sabha. Now it's time to request Chairman, Department of Chemistry, Professor Dr. Mia Habibur Rahman Mahmood, for a vote of thanks.
Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Thank you very much, Dr. Ishtiaq Ahmed Khan sir, जो आज के स्पीकर हैं. I'm really thankful for his precious time. He actually spared his precious time for a really wonderful talk at Department of Chemistry, University of Education, Lahore. आज की इस टॉक से हमें जो कोविड-19 है उसके जो रीसेंट ट्रेंड्स हैं और फ्यूचर चैलेंजेस हैं उनका पता चला. आई होप कि हम इस जो इंफॉर्मेशन शेयर हुई इनको हम ध्यान में रखेंगे और ना सिर्फ ये हमारे लिए बल्कि फ्यूचर में जितनी भी हमारी टॉक्स होंगी ان کے لیے یہ ایک کی رول پلے کریں گے. ویبینار کے آج کے جو آرگنائزنگ ہیں ڈاکٹر محمد اسلم صاحب اور فیکلٹی آف ڈپارٹمنٹ آف کیمسٹری. آئی ایم تھینکفل ٹو آل آف دیز کہ انہوں نے اس ویبینار کو آرگنائز کیا. اور ہماری خواہش ہوگی کہ ڈاکٹر اشتیاق احمد صاحب کو پھر ہم انوائٹ کریں کسی وقت، کیونکہ یہ ٹاپک امپورٹنٹ ہے، ریسنٹ ہے، اور ان کی جو ماشاءاللہ جو انفارمیشن انہوں نے شیئر کی ہیں، جو ریسرچ شیئر کی ہے، آئی بلیو کہ یہ باقی یونیورسٹی کے ڈپارٹمنٹس کے لیے سب کے لیے بہت ضروری ہے تاکہ ہم سب کو یہ انفارمیشن فلوٹ کر سکیں. تو فزیکلی جو ہماری نیکسٹ اگر کوئی ہوتی ہیں ویبینار وغیرہ، تو ان میں ہم ان کو پارٹیسپیٹ کروا سکیں اور ان کا لیکچر جو ہے وہ ہم دوبارہ رکھ سکیں. اگین تھینکفل ٹو ڈاکٹر اشتیاق احمد خان صاحب اینڈ آرگنائزنگ کمیٹی. تھینک یو ویری مچ.
Welcome, sir. Thank you so much, everyone, for joining this webinar. And Allah Hafiz.
Thank you very much. Allah Hafiz.
Allah Hafiz, sir.